In this presentation, our focus is going to be on the portion in the orange, mitosis and cytokinesis. Mitosis is sometimes called karyokinesis. It's specifically referring to division of the nucleus, which is the first step of mitosis, and then the second step is called cytokinesis, which is division of the cytoplasm into two separate daughter cells. We're going to look first at mitosis, the division of the nucleus. It's divided into 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. And I find it's easier to remember these if you know what the term means. So pro means first, meta means middle, so it's the middle phase, but also something that's going to happen in the middle of the cell that's important. Ana means to separate, so something's going to separate during anaphase. And finally, telophase, telo means the end. So now we're going to go through each of these phases, beginning with prophase. In prophase, the nucleus is going to break down, sort of dissolve, and then the other organelles are going to get pushed to the edge of the cell. The nucleolus, which was that dense region inside the nucleus, disappears as the chromosomes begin to condense. In animal cells, there's these centrosomes that are going to migrate to set the poles and set up a network of fibers called spindle fibers, and the chromosomes are going to begin to condense into these very compact shapes. At prometaphase, the chromosomes have condensed into the structure we see here. You can see there's what we call 2 sister chromatids. They represent the copied chromosomes. They're held together by a centromere. And then there's a protein here called the kinetochore protein that's going to attach to the spindle, and then the spindle can start to move the chromosomes around the cell. As the chromosomes are moved around the cell, they are going to form in a line in the middle of the cell. It's called the metaphase plate. And this is why we call this metaphase. The chromosomes are lining up in the middle of the cell. There's a fiber from each side of the cell connected to each chromosome. Once the chromosomes are lined up, a signal is sent to release the 2 sisters so that the chromosomes then begin to migrate toward opposite poles. So each chromosome, one copy is going to go towards each pole, so that each resulting cell is going to have a complete set of chromosomes. And then finally, telophase is kind of the opposite of prophase. The chromosomes have reached the poles and begin to unravel, decondense. The nuclear envelope begins to reform around those chromosomes, and we have 2 new nuclei inside the cell. The spindle has broken down as well. Now that the nucleus is divided, the cell itself can divide, and this process is called cytokinesis, resulting in separation of the cytoplasm into 2 what we call daughter cells. Because of the physical characteristics of the cells, the process of cytokinesis differs when you compare animal cells to plant cells. In animal cells, because there's no cell wall, it's just a membrane that's very flexible, there's going to be a network of proteins between the 2 nuclei that are going to constrict the cell, we call a cleavage furrow, and it's just going to pinch the cell until you get the 2 membranes to fuse and separate into daughters. In plant cells, the rigid cell wall will allow the cleavage formation. So what we see in the plant cells is a cell plate formation where the membrane forms between the 2 nuclei and then extends until it separates the 2 cells into separate